Hello, everyone. Welcome to today's Research America Alliance member web meeting. And thank you for being Alliance members and for joining us today. I also want to extend my gratitude for your joining us today on Juneteenth, which this year comes as a time of reckoning and reflection across our nation. And we at Research America are inspired by the ways in which so many of you have been moved to listen, to learn, and to take action to address the need for racial justice now. Last week, we posted some of the resources we've been exploring, and they're linked in the chat box now. Before we get started, I think you know the drill, but there are instructions on the screen to mute and unmute your line. Before you type your questions into the chat, we'll be saving those for the end. So just go ahead and type them at any time. And my colleague Terry will be serving as our questioner of Jeff. And for those of you dialing in by phone, we'll open up the phone lines after the discussion, and you can hit *6 to unmute your line. So our guest speaker today is Dr. Jeff Allen, President and CEO of Friends of Cancer Research. Friends of Cancer Research is an advocacy organization driving collaboration among partners from every healthcare sector to speed life-saving treatments to patients. Jeff has been with the organization for more than 10 years, leading the discussion on many issues related to the FDA, regulatory strategy, and healthcare policy, and many other things. So, Jeff, thank you for joining us today. Over to you now.
Great, thank you. Thanks, Jenny, and to Research America for hosting these venues generally, but in particular for inviting me to talk a little bit about some things that we've been working on recently, and thanks for everyone who's joined as well. I guess if we can just go to the presentation, that would be great. What I thought would be hopefully helpful, at least in terms of starting a discussion, would be to spend a few minutes today giving a couple opening remarks around some of the impacts to biomedical research and clinical trials generally, and hopefully transition that into a summary of a program that we've been working with the Reagan-Udall Foundation for the FDA, as well as the FDA and a growing number of stakeholders to try and launch around using real-world evidence as an additional research tool. So to go to the next slide and start on clinical trials, I think, obviously for all of our organizations, the research enterprise generally, if not the world, planning ahead for a pandemic it has been a huge challenge. So there were a range of various immediate impacts that I remember happening very quickly in the early March timeframes as things started really accelerating in terms of a rapid response and how to begin to try and contain the spread of COVID-19. And in the research side, one of the most immediate things that I think was observed was a number of clinical trials, both from research organizations or commercial sponsors, halting the trials altogether as part of their strategy for dealing with COVID-19. And this was done for a variety of worthwhile reasons, including things like protecting patients from high-risk environments, with many patients having to go to a trial site, and those trial sites being the same types of hospitals that were setting up their own processes to try and deal with COVID-19 generally, and how to prioritize the safety of the healthcare workers, responding to those patients that were infected with COVID-19, as well as thinking about how to reallocate resources. And unfortunately, perhaps at that time, the clinical trials infrastructure had to be temporarily put on hold to be able to respond to those immediate needs of the healthcare system as a whole. And what that happened, no surprise, based on that, was a very sharp decrease in enrollment that was seen. I cited one stat on that slide there that was from a large-scale analysis conducted in large part by Medidata that showed that from comparing this year March trial enrollment to last year's March trial enrollment, depending on the indication, that for most major disease areas, there was over a 50% reduction in participation in clinical trials, some being even higher than that, up into the 80th percentile. But this had ramifications downstream and continues to, in terms of things like the completion of those trials, or even what the patient's access to experimental medicines could be, opting to stay out of a healthcare facility for their own protection caused many people to start thinking about what they might be able to do at home, as opposed to potentially enrolling into a trial where they would have to be more regularly going into those healthcare facilities. And I will say that it was very quick that regulators and government officials did respond to acknowledging this as a stressor on the system by issuing rapid guidance, at least for consideration for those that are conducting and designing trials, about how to respond and report things like changes in protocols, or what to do with missing data because of dropout based on the risk of continuing on the trial, or having to collect data in a different way. How to be able to still gain the necessary information to complete some of those potential trials. But in addition to trials, other things that are continuing to still recover from this are things like access to routine screening. We certainly have seen this in the oncology space at very high levels, with some studies pointing to routine preventative screening plummeting almost to the tune of almost 90% in some instances. And what's likely, in addition to some of those preventative screening, even in terms of diagnostic testing that may have to be performed at a clinical laboratory, those are likely to also have been affected. If you go to the next slide, I think some of these immediate things that have been observed over the last couple of months and hopefully are coming back online, there are still things that need to be considered for the long-term maintenance of the biomedical research infrastructure. And some of these are unknown due to the novelty of the coronavirus in itself. As things continue to evolve, we learn more about the manifestation of the disease and the long-term effects of it. And some of these questions still remain to be answered. Things like, will a patient who's had a severe outcome associated with COVID-19 be immune compromised further? Will they be able to mount the same immune response in response to treatment? In cancer, rheumatoid arthritis, other types of diseases that rely heavily on some of these biologic modulators. And in terms of the clinical trial design, some of this is still being worked out too. If the former is true, where some of these patients may need to be accounted for differently, how do you build that into a trial? Do patients that have recovered from COVID-19, for example, need to be stratified to be accounted for in testing a new medicine in order to make sure that those results aren't skewed by the population that may have a different performance because of their exposure to COVID previously? And that may have long-term effects, and we've seen this in terms of other infectious diseases, at least in the oncology space, where even now just loosening things like eligibility criteria, as there's been a better understanding for things like HIV, that many of these patients can be included in regular oncology trials. And I think it will take some time before recognizing whether that can or can be across the board true for COVID-19 patients, or if there needs to be different strategies put into place. But this is significant, particularly in oncology, due to the overlap of these patients. Several of the demographics of cancer patients are similar to that of oncology or of COVID-19 patients as well, in terms of their age demographics, comorbid conditions, pre-existing conditions, both of which place individuals at higher risk. And in terms of where the focus will be, I think we'll continue to see shifts in priorities from all sectors. Those that are performing the trials, investing in the trials, and figuring out how to move them forward, I think is something that will be for the long term. But on the next slide, I think there are a lot of things that can continue to be potential opportunities to learn from. Perhaps some of the things that have been designed to get new COVID-19 trials up and running can be held onto and applied more broadly. Things like expediting IRB reviews, allowing for more advanced planning of modifications and trial amendments, which can slow trials down. By keeping some of those practices in place, perhaps it will help clinical research more generally. We've also seen a renewed increase of interest in the utilization of things like master protocols, which can streamline enrollment, expedite the launch of new trials by having more of a freestanding clinical research network that can be used to deploy data onto new questions, as opposed to having to start from scratch each time, which is being actively deployed by the NIH and others in terms of researching COVID-19 therapies. And in addition to some of the challenges about patients having to go toward their trial site, a lot of different strategies are being deployed to allow them to be able to stay at home more, participate in consultations with healthcare professionals remotely, even having their medicines delivered to them at home where that's possible. And that's something that might be able to, in the future, allow more individuals to participate in clinical trials into the future, should those processes be able to be maintained. But on the next slide, I wanted to spend sort of the second half of our discussion here talking a little bit about one way that we have been working with a large number of stakeholders to approach this situation a little bit differently. And that's in the utilization of real-world evidence. So a couple of weeks ago, in partnership with the Reagan-Udall Foundation and the FDA, we launched the COVID-19 Evidence Accelerator. And this is a multi-stakeholder collaborative project involving numerous different organizations, healthcare systems, government, hospitals, data aggregators, to try and come together and perhaps in a more organized way or a coordinated way, try to address some of these really rapidly emerging questions, with the goal being that while numerous different organizations have a huge capacity to do this type of research based on electronic health data, could combining these efforts, particularly around a common set of questions, be able to create larger data sets than if any one organization was doing it itself. And in order to get to that larger number, be able to develop the evidence faster. So the next slide, please. What this is meant to do is recognize that there are a great deal of existing efforts underway. Those at the FDA in particular, like the Sentinel Initiative, which has a long-standing capacity and experience in running these types of large multi-participant data aggregating studies, as well as other government entities like the CDC or the VA or the NIH, as well as other organizations like PCORI that have had different functions in terms of creating research networks, but trying to bring them together, coupled with information from the private sector, and various different data sources that may not have otherwise been leveraged in some of these electronic data efforts, to sort of bring them all together in a coordinated way and create a larger real-world evidence type community. So next slide. So the way that we've envisioned this, by having creating a organized approach toward bringing all of these different parties together, has really been to start at the beginning and bring the community together around prioritizing various research questions that could be answered jointly in a more rapid fashion. And by aligning on things like common data elements and variables that could be collected, and then further aligning on the analysis plan, we've constructed something that can be a very distributed type project, where the owner of the data does the analysis in a common way on their data without having to move that data. And while aggregating and pooling data is a powerful tool, being able to allow that data to stay where it resides and still perform a common analysis can provide some strength by confirming repetition, essentially, to increase confidence in those results. So one work stream of the Evidence Accelerator is what we've called parallel analyses, with this idea that multiple different organizations will help design a common protocol, apply it to their data, bring the results back together in order to confirm each other's finding. This is based on some pilot work that we've been working on over the last couple of years in oncology, and that work stream, independent of COVID, is continuing but it's demonstrated this idea that when data from different sources are able to apply some common methods, you can show repetition and finding, but it also allows for greater transparency into the methods and greater participation from additional networks to be able to participate in these research studies, which speeds things up. In addition to these types of parallel analyses, even just having that community approach by simply providing a forum where organizations that otherwise may not be connected to each other can find out what people are doing. So we call these our weekly lab meetings. They're Thursday afternoons where different organizations, there's usually just a couple of presentations each week where people share some of their intermediate findings. We call it a lab meeting because that's very much what it's like. Designed to be an informal venue where people get together with their peers, share some of their results, ask questions, exchange ideas, perhaps create collaborations to move forward faster. So it's not as formal, it's not meant to be formal as a scientific conference or some sort of publication venue, but it still creates that peer-to-peer interaction and has already led to a number of different linkages. And in addition, we're beginning to look at where we might be able to expand this approach to other areas. On the next slide, you can see sort of the rapid progress that we've been able to instigate from the beginning. So starting at the end of March, we really tried to reach out to nearly 50 different research type organizations, different health systems, public, private, to do a bit of a landscape analysis about, number one, a set of common key questions that were identified, excuse me, that were identified as things that might likely be emerging in the COVID-19 landscape, both immediate priorities and longer term, to see if we could identify common areas of interest, as well as key data elements that would be needed to answer those questions using real world data. And that fed into the parallel analyses. But what you can see here is that we very quickly put that landscape analysis together, realized at that point we needed to have a venue to keep all of these organizations together so that they could share their ideas, in addition to launching some of the research projects. So on the top, you can see in mid-May, we launched the series of lab meetings, or in mid-April, we launched the series of lab meetings while we were preparing the first parallel analyses. In mid-May, we launched Project 1, which was related to the utilization patterns of hydroxychloroquine as an initial pilot, which is to be completed in the next couple of weeks. Since that time, just in the last couple of weeks, we began to bring people together around additional subjects, such as the use of diagnostic testing. And obviously that's of growing importance. While therapies are incredibly necessary in order to help people that contract COVID-19, the utilization of diagnostic testing will be important to be able to manage it into the future. So both in terms of the diagnosis of the disease, which was the immediate focus into the pandemic, but now growing so into things like serological antibody testing to be able to potentially detect those that have recovered from the disease and potentially, hopefully, have some degree of immunity against reinfection into the future. On the last slide, you can see how this all is sort of working together in these various different work streams. So there's the therapeutic side of the evidence accelerator, which has the weekly lab meetings that I described, the parallel project, the parallel analysis project, number one, focused on hydroxychloroquine, but already in discussion about moving to other areas of research about drugs that are out there, some of which have had initial information presented about them, to be able to move those into larger settings to conduct the research, the expansion into the diagnostic work stream, which is second there, and then the potential maybe even to the future when you think about hopefully soon when we'll be talking about distribution of vaccines, how to have a post-marketing infrastructure already in place to be able to conduct the necessary long-term monitoring for safety and effectiveness of those strategies into the future. And then finally, how different work streams can sort of cut across that. Like I mentioned, the increased prevalence of COVID-19 in cancer patients, we've been working with the FDA Oncology Center of Excellence to also begin to identify priorities that they're seeing in cancer clinical trials and drug development to be able to think about how some of this real world evidence could help inform their approach to developing and regulating drugs into the future. So I'll stop there. I'm happy to take any questions and send things back over to Terry and Jenny.
Thanks, Jeff. Wow, terrific work. I'm going to start off with a couple of questions, and then I think we have some from our participants. So yesterday we had Dr. Ned Sharpless on a webinar, and we were talking a little bit about the active effort at NIH, which is also attempting to bring stakeholders together around different research processes and methods. How are you guys interacting with that group, and how does your effort fit into what they're doing?
Yeah, we've interacted with a number of the different participants in the active study, really on a granular level too, conceptually on how we can sort of leverage each other's efforts, largely with active being focused on developing new therapies. And what I think the Evidence Accelerator can contribute to that is for longer term, post-market, real world, once it's in a broader population, monitoring of those successful therapies. But in order to do that, ideally, we'll have mapped out some of the appropriate common data elements so that we're able to measure things in a similar manner, and have had a number of discussions with the Foundation for the NIH and others that have been working on that. So at least we're aware of each other's efforts and try and leverage them and make sure there's that open communication, which is pretty much what this is all about, creating more open channels of communications between different organizations that may not be regularly interacting.
So your work really is more focused on the post-market setting.
Yes, in large part because a lot of the data that's being collected and contributed through the Evidence Accelerator are things from electronic health records, because those are most immediate. I think we'll start more addition of information from insurance claims, for example, which might have a little bit of a lag time in some instances. But where I think this provides a unique snapshot on top of those original clinical trials is by having a more broad-reaching, diverse, geographic, individually, different population to be able to look at the extent to which some of the early clinical trials hold true once it's in a broader, more diverse population and over time.
Terrific. Thanks for that clarification. I think we have some questions from our audience. Terry?
Sure, Jenny, we sure do. And just a reminder to folks that you can click on the questions box and type your questions in there as is shown on the screen. One question so far, can you provide, Jeff, some examples of the kinds of data being collected using these parallel analyses?
Yeah, so it's an extensive set of variables, which range from demographic characteristics around, different age brackets, ethnicities, sex, comorbid conditions, other concomitant medications, but also different analyses that are fit for this particular circumstance. Even things like time on ventilation, time for ICU stays. Different measures that would be very important around really trying to elucidate where some of these therapies are providing benefit. I think what we've learned very quickly is that the methodological approach and the transparency of those and really aligning on those are critical from the outset. And given the particular challenge with COVID-19, both in terms of its novelty, but how it affects people in a wide, diverse setting. So not everyone that's infected ends up in the ICU. Not everyone that's infected even develops symptoms. So to try and parse that type of the population presents its own challenges because it impacts people so differently. So trying to isolate that, those subsets of responses in real world data has sort of been a early challenge to then be able to ask the questions and account for that in the analyses. Given that in some of these early treatments like hydroxychloroquine or remdesivir, or even some of the information that has recently been presented around dexamethasone is, oftentimes as people are utilizing these drugs in practice, it may be given first to the sicker patients, where the patients and the physicians are looking to provide something. So even accounting for those differences in the population is something that's been of great focus in setting up some of these protocols.
Jeff, I'm going to ask you a question on the diagnostics front. Clearly the really smart use of diagnostics is going to be key to managing the pandemic better and, getting us all back to some new normal. What are some of the issues that you're looking at in the post-market space to help us with that?
Yeah, so this has been an interesting approach even right now, because unfortunately the way that the greater healthcare system is set up, this information isn't readily linked together in most instances. And the priority in terms of the tests may be a little bit different. And by that I mean the characteristics of a test to diagnose an individual who has COVID-19 may be a little bit different than the properties you would want to see from a test for showing that someone has potential immunity to COVID-19. So for example, a test that it gives a false positive for someone that that tells them they have the virus, but they actually don't, that may not be present as large of a risk, because they'll take precautions, monitor their health, social distance, and be able to try and prevent the spread of the disease. So the reaction will be precautionary. But at the same time, a false positive of a test showing that someone has immunity would present a higher risk, because that individual might go interact more with different individuals thinking that they have immunity, but that false positive means that they actually don't. So making sure that you minimize the false positives to the fullest extent, for example, on the serological testing, may be of higher priority than it necessarily needs to be for the original diagnostic test. So I go into that just to say that, like, even gaining this information on each individual test that's out there is really important. So in the real world setting, what we're starting to think through is, while a lot of these tests, even if they've gone through an emergency use authorization, have some preliminary performance around that negative predictive value and positive predictive value, from an analytical standpoint, as part of their movement into market, what the hope with real world evidence that we'll be able to put a system together that will show, well, we may know the number behind on a very limited set of clinical samples of negative predictive value, but to those individuals, to what extent does someone who is told you test positive for antibodies to COVID-19 actually get re-diagnosed with COVID-19 or get admitted to the hospital with a diagnosis of COVID-19, indicating that perhaps the performance of that particular test, doesn't match its analytical performance that had been shown initially.
Really important. I mean, you're getting at a lot of the questions that have been raised. I think we have another question from our audience, Terry.
Yes, we do one or two more here. One is, are there steps being taken to ensure that the data that is collected is representative of a diverse population? As we all know, COVID-19 has impacted communities of color in a really significant way.
Yeah, really important question. And I hope that what we'll be able to do by bringing these large data sets together is even add to that body of information. That there's certainly been a very conscious effort to be able to collect on all of these fronts, the exposure to the different drugs, the access to the testing, the outcomes associated with COVID-19, to further that evidence base to help strategies into the future to begin to focus on those communities at higher risk, or that may have lesser access to some of the, particularly on the diagnosis side, to be able to deploy efforts and provide at a granular level, what are the quantitative numbers behind that? So that's been something that we've tried to incorporate into all of these different protocols to be able to measure that.
Thanks, Jeff. I think we're going to need to move on, but if we have Alliance members on the call who would like to explore the Evidence Accelerator further, is there a place on the website where they can reach out to get more information and possibly get involved?
Yeah, definitely. So with the Reagan-Udall Foundation, we've recently launched evidenceaccelerator.org as an informational page that should have the ability to sign up for updates or join some of these meetings. There's information on the Reagan-Udall Foundation website, as well as ours at Friends of Cancer Research. I think it's /COVID-19 to find information. Summaries of these different meetings are accessible, and ways to be involved, and we'd welcome that. And don't hesitate to reach out to me too. But
Thanks, Jeff. Well, this is exciting. You guys are really going to make a huge difference, particularly as the FDA, starts to prepare its guidance and helps to, further establish real-world data, real-world evidence principles going forward. So terrific. Thank you so much.
thank
Have
you.
a good weekend.
Thanks so much. You too. Good to talk with you.
All right.